presentation. Okay. Do you see it, Lei?
Yes.
Okay, great. Then I will start. Great. Okay, cool. So the topic of today's lecture is doing open science, but it could also be doing good science, and you will hopefully at the end of the lecture all know why. So first I wanted to give you a little bit of information about my research, even though Lei already introduced me very nicely. So I did my master in Vienna in clinical and biological psychology, and I focused on social pain, so being excluded or included, on concepts such as self-other distinction and emotional egocentricity. And I investigated this in the domain of autism spectrum disorder. And then after that, in 2017, I started my PhD with Klaus Lahm at the SCAN unit. And as Lei already said, I'm investigating mainly empathy for pain. I use functional magnetic resonance imaging in most of my experiments, but also a lot of electrical pain. And recently, I've also expanded my research to pro-social behavior. I use placebo, or I investigate placebo effects. And I'm very interested in interoception. But this is just on the side, so you know a little bit about me. I had a quick look at the results from the survey that we sent out to you. And I just checked again, and we have one response more. So the 25th response is missing now. But just so you get an overview, it was very good to see that although some of you have heard of the concept of open science, there is still something that I can tell you about it. So half of you have heard about the concept, but the other half has never heard of it. So it's nice that I can still hopefully teach you something about it today. And if we look at the current knowledge on the terms that I gave to you, it's also interesting that although words like data fabrication and data falsification and the replication crisis are already known to you as a group, although it's quite varying, some of these terms like harking, p-hacking, file drawer problem, you also don't know so much about yet. So this is another evidence that it's very good that we do this lecture. Now already for the second time. I already did it last year, and I thought that students seemed very interested in it, and I think that's nice. Yeah, also if we look at the methods used in open science, like pre-registration, registered reports, and so on, most of you have never heard of this, so that's very nice. So here's a short overview what we will talk about today. I'll start with some basic concepts that might be relevant if we talk about open science. I'll then go on to the replication crisis, what it is, and what other concerns there are in science at the moment or that were there in the past. I'll then go on to talk a little bit about the open science movement, how it evolved, and what the principles are. And then we will do a short why or why not do open science, and talk about some specific solutions that open science suggests to the problems and concerns that are present in science still today. And in the end, we will finish with how you as students can already become active now. Just to give you a heads up, so there are two kind of interactive elements. You can ask questions in the chat, and I will answer them in short breaks after each of the topic blocks. And then we will also attempt to do a live poll over Zoom. I hope that it works. We tried it before, so this will be about good or bad scientific behavior, but it will come later. Good, so I'll start with some basic concepts. I know that you got this figure already in one of Lei's slides, but I just wanted to have it again. So we have alpha, a type 1 error. That basically means we say that something is there when it really isn't. And then we have the type 2 error, which is the beta, which means that we say something is not there, even though it really is. And those two errors are related. Alpha is related to the p-value. So how likely the pattern of results that we observe in an experiment is, given the assumption that the null hypothesis is true, so that there is no true effect. While beta is related to statistical power. So power is the likelihood that a study will detect an effect when there actually is an effect. So that would be 1 minus the beta. Then there are two other terms that are relevant for the lecture today. One is called researcher degrees of freedom, and this has to be distinguished from statistical degrees of freedom, which is something that is relevant when we do things like t-tests, for example. But researcher degrees of freedom means something else, namely the concept that researchers have a lot of flexibility when they design a study, when they analyze a study, when they do an experiment. And when they report the experiment. So degrees of freedom basically means there are a lot of options that researchers have to do a study and to report a study. And then there's also a big difference between two types of hypotheses that you maybe already heard about, confirmatory versus exploratory hypothesis. While confirmatory means that we have a hypothesis and we want to test this hypothesis. So a priori, before we do the study, we have an hypothesis and then we do the study to test it. So that means that the hypothesis is specified before the data collection. While exploratory hypothesis or hypothesis generation means that by doing a study, we try to come up with hypothesis a posteriori, so afterwards. Based on already examining collected data or looking at data that you already have. And this is a very important distinction if we look at later at some of the concepts or the things that open science proposes we can do to make research better. Okay, so we already do the poll. So let me try to, I think I have to unshare and then I here it is, yes. Okay, I will start the poll and hopefully you will all see it. And then you can answer the questions. That are there. And we'll have a look at them later to see what your results were. So the questions,
a little bit more background. All those questions are about what do you think if the behavior is acceptable in terms of good scientific
exactly.
And all those answers are yes or no, and maybe, and don't think about too much. Just give your immediate feeling.
Yeah,
regarding
exactly.
How do you think they are?
Yeah, and I'm doing this because later we will look at the same statements again. We will see maybe your opinion has changed. So keep in mind how you answer now, just spontaneously to the questions, and then we will have a look at them later. Okay, I have one answer logged in. 2. Yeah, so I mean, we have no's, but we also have some maybes. That's interesting. And I'm not saying that all of these statements are a definite no. I mean, I of course included some maybe not so clear examples. Yeah, let's just wait a bit longer. We have nearly half of you who filled it out already. Okay, 17 out of 24. Yeah, I mean, I'm seeing the answers now as you put them in, and they are already quite diverse, which is very interesting. Some are pretty clear in one direction, but others are very different. Okay, we're missing 5 more people. Let's just wait a bit longer and then we can go on. If you have any questions already now, don't hesitate to put them in the chat and then I will look at them when we have a break. Okay, 5 more people. Ah, yeah, 20. So 4 more people. Mobile, I don't know actually. We didn't try it. On the, maybe you can try if it works. Otherwise, we look at, well, then probably not. I only tried it on a computer. Could be that it doesn't, because it's a pop-up window, it could be that it doesn't show up. But we look at the statements later again, so that's fine. We can also, you can evaluate them later. Maybe it's even better because then you have more knowledge. Okay, missing 4 more people. So everybody who hasn't logged their answer yet, please do so. Otherwise, I will stop the poll in a minute. Okay, I guess since it's not changing anymore, maybe most of you are done already. So I will, yeah, maybe if you are still working, write in the chat so I don't close it when you're still going on. But since I haven't seen a change in a while, I thought I'd stop it. I think it's fine, Leno, to stop it.
Yeah, okay. Mm-hmm.
Okay, great. So I'll stop. And later we'll have a look at the results. So don't worry about them now. I'll share again the screen and I open my presentation. Great. Okay, so let's go on. Next up, we'll talk about the replication crisis and some other concerns that science has. So some of you already heard of the replication crisis, as you can see here on the right side. So 20 out of 24 have heard about it or are familiar, which is nice to hear. But just to give a short overview what it is and how it evolved. So if you ask scientists or researchers, there's actually also a very big consent that there is kind of a crisis, as you can see in the left image. But there's also some people who say, well, a slight crisis or not really any crisis, or they're not sure. So it's very diverse if you ask scientists. And I just wanted to give you some examples of studies that couldn't be replicated, meaning that a study is done that shows a certain result, and then it's conducted again the same way, and the authors look if they find the same result again. And the reason for this replication crisis started with a lot of high-powered replication studies. So a lot of studies that tried to replicate previous findings, but then they couldn't. So the replication rates were very low. And also with this, the outcome was that there was an increased awareness of good and also bad scientific practices. So while all of these studies were not replicating, people started to wonder why. But still, a lot of researchers disagree about the extent of this problem that is called the replication crisis and the solutions to this problem. So here are just some examples. For example, there was a study that in 2010 that showed that if you adopt a power pose, so like in the picture, if you stand up very tall and you put your arms on your hips, that this will make you act bolder. So they found that the participants who stood in one of those power poses had higher testosterone, lower cortisol, and they made riskier bets compared with participants who hadn't stood in a power pose. But this was never replicated. Another example is that smiling will make you feel happier. So in 1988, this was a study that participants found cartoons funnier when they held a pen between their teeth, like in the left picture, compared to when they held the pen in their mouth, like on the right side, where they didn't smile. And this was also not replicated. And then this is the most funny example, I think, that a study found that if you study for the exam after you did the exam, you can improve the performance on that exam. And this is, of course, not possible since, yeah. But this was, of course, also never replicated. And all of this, these are, of course, some examples. But in general, the failure to replicate previous results led to something that we call replication crisis now. But there were also other concerns about the integrity of science in general that came up with this whole, why are we not able to replicate results that we already found or that others already found? And this is where we come to the so-called QRPs, or questionable research practices. And this is like an umbrella term for a lot of different terms under it, one of which is p-hacking. So we already talked about researcher degrees of freedom, so how many options researchers have to do an analysis, to report a result, to do a study. And p-hacking also, because of the p, refers to the p-value. In the poll that I did before, actually a lot of you got the correct definition of a p-value, which is very nice. But p-hacking therefore means that you try to get under this p equals 0.05. So this is the magic number, and especially in null hypothesis significance testing, so the usual statistical test that people do, or the framework that people do them, the p-value is very important. And what do you do if it's not significant? So if it's above 0.05, this is very bad, of course. So there is this trend of just doing lots and lots of analysis until you find a p-value that is above, or that is the threshold that you want it to be. And this is called p-hacking. So you exploit the researcher degrees of freedom that you have to get statistical significance. Phishing is a similar term, basically just meaning you're trying to fish for until you get a statistically significant result. And importantly, not only that, but then also the secrecy that comes along with it. So you not only do lots of different analysis, change small things about your analysis, but you also don't tell anyone, because in the paper, this would sound really bad. So in the paper, maybe it just says we did this one analysis and it was significant, even though all of the other 20 analyses that were done before are not reported. Another questionable research practice is post hoc theorizing, that is very closely related to harking, or hypothesizing after the results are known. And this means that when you have the result that you found, you did some analysis, but this was not what you hypothesized. So maybe you had a different hypothesis before, but you found something that is interesting, but it was not, it's maybe the opposite of what you wanted to find. What is been done, or what is being done sometimes, and this is something we don't know, but that is hypothesized, is that people write, I wanted to research, like this was my hypothesis all along, even though it was not. Or in a more subconscious way, when they find a result, they tell themselves, yeah, I mean, this makes sense. This is what I wanted to find in the beginning. So that's also a questionable research practice, just because it's not being transparent. So we thought something else before, but now we're telling ourselves, or we're telling others by writing it in the paper, that we thought something else was true. Yeah, about this I talked already. So there's a lot of flexibility in how we do an analysis. If we look at already the different statistical programs, so R, SPSS, MATLAB, there are so many ways to choose a program. Then how to do the analysis, so what function, what underlying function to use, that all of these analysis choices can lead to different results, actually. One of the problems is also the inappropriate use of statistics. And finally, what is also a big problem is that publications are not always free to everyone. So there are journals that have open access. That means that everybody can read the papers that they publish. But there are also journals that don't have that, that have a so-called paywall. So you have to pay for each article. And this way, not everybody gets to see all of the results. And this is very bad if we look at the fact that we need all the knowledge we can get. If, for example, we research in a certain field, we want to know about all the papers that happened. But if we don't have access to the publications, how will we incorporate those in our own studies? With this lack of access to publications goes along that we also don't have access to lots of data, material, or code that goes along with the studies. So the default usually is that you write a paper, you publish it in a journal, but that's it. You don't give any access to the raw data that you collected, or to the material that you used in the study, or to the code that you used to analyze the study. And I'm maybe exaggerating here. Of course, there are journals or there are authors who do this regularly already, but this is also still something that avoids us getting all the information that we need. And then, of course, let's say on the very negative end of the spectrum of questionable research practices is things like fabrication. So we just invent data, falsification, we changed it, or fraud. So this I put on the list anyway so it is complete. This is also an image that Lei already showed you, maybe in a little bit different way, but this is just the scientific process or the circle where it starts with generating a hypothesis, designing a study, collecting data for the study, and then analyzing it, interpreting it, and then publishing it. And in this research cycle, a lot of things can go wrong. So if we, for example, look at the lack of replications. So the hypothesis and the results that were found cannot be replicated. Then lots of people design studies that have too low statistical power, for example, low sample sizes. This can also lead to results that might not be what they would be if we had used bigger sample sizes or had higher power. When we collect data and analyze data, we can engage in p-hacking, also in interpreting. And then when we interpret the data, hacking can be a problem. So we find something and we say, yes, this is what I wanted to find all along. It makes the story very nice. Let's just forget about the original hypothesis that we had. And then finally, there is a lack of data sharing in the publication process, and also publication bias that I will talk about later. So in general, about what we have heard so far, there is a desire in scientists or in science in general to better understand the choices that are made throughout the cycle. So some of these things are not very transparent. People just don't know what a researcher does until the study is published. So this is kind of the start of the open science movement. But before we move on to talking about solutions and what the open science movement is, I will give you an example of p-hacking, because I think it's a very funny example. So I'm here in the p-hacker app that you can also have the link on the slide later. You can go on there yourself. And I'll just open this again. So let's say we want to investigate whether listening to classical music improves short-term performance on certain kinds of mental tasks. This is called the Mozart effect, also something that was published and found in the past. And let's say we have 20 participants per group. So we want one group who is going to listen to classical music, and we have another group that is going to be the control group that doesn't, or that listens to another type of music. And let's say that in reality, there is no true effect. So the Mozart effect does not exist. It doesn't improve mental ability. And because we want to be really sure that we look at all different angles, we use 10 different mental tasks. And now we go back to the app. So I have the Mozart group here, which is our experimental group. I have the control group here. And then we're going to do 20 different participants. In reality, there's no effect. So there's no such thing as the Mozart effect. And then because we want to be really sure, we do 10 different tests. So 10 different tasks that we compare our participants on. So let's run the experiment. Okay. So as you see here, if you look at the column with the p-values, nothing is significant. And everything is really far away from 0.05. That is super bad. We cannot publish this. So maybe let's just explore the data a bit more. So if we go down here, we can look at the different tests that we did. For example, this one. And if we look at the distribution, so these are the data points of the subjects, and this is the control group, this is the Mozart group, we see, well, there is a trend of the Mozart group to be a bit lower on this test than the control group, but it's just not significant. They are too close to each other, the means. So what could we do if we would want to p-hack now? So we see that this point is very far away from the others, and also this point. So if they were outliers, we can just exclude them, right? So let's click on them. This one is gone. Let's exclude the other one as well. So now we have a much nicer distribution, right? So if we now look up here, finally, we have a significant result on this task that we just excluded our outliers. That's nice, right? I mean, and if we're still not happy because we would like some more, results in the others as well, we can, for example, add some new participants. So let's not do 20 per group, but let's do some more. But now this on the top here has disappeared. That's interesting. But others, we have some trends here. So of course we could go on and on here, but this is just an example to show you, and you can try this out yourself later, how many different degrees of freedom you have in a study like this by just comparing two groups on something, and how many different options you have to find significant results. And this can be very bad if used like this in studies. So here are some other reasons why there is a replication crisis. I mentioned already publication bias before. And this means that there is a system in place in journals, in scientific journals, where only selective papers or selective results are being published. And again, I'm exaggerating a lot, but it is like that only or mainly novel and fancy results that have significant p-values and have a very good story in general, those are being published in the very good journals with high impact factors, while other studies that don't have significant results or that are maybe not so nice from the story are either being published in lower journals with lower impact factors, so they don't have that big of an impact, or they are not published at all. And this leads also to the file draw problem. And this just describes a situation where studies that have null findings, so no significant results, they are just never published. They just land in a file drawer of the scientist and they stay there, and nobody ever knows that this study happened. And this is a really big problem because the studies that are out there and the studies that are being published are not a good representation of all the studies that are actually being done. And this is something that open science also tries to tackle, as we will talk about later, that this should be changed. In general, the incentives that scientists get for publication are also kind of wrong in a way. So I said before, flashy hypotheses are always very good. The results should at best be novel and significant. Also, more studies are better than lesser studies, but better quality studies. For example, if we have 10 studies that have a sample size of 20 people, so we have 10 papers out of it, compared to, let's say, one study with 200 as a sample size. Of course, the latter takes much longer to collect the data and maybe longer to analyze. But then you just have one study in the end. While if you just do very small sample, low-powered studies, you get more publications. And this is unfortunately something that is still favored a lot, having a lot of publications and publishing very quickly, especially when you're doing a PhD and when you're just starting out in science. So I will take a short break to look if there are any questions. As I said, I think there are. You can ask them anytime. But I think so far, yeah. I once heard in a lecture that there is a journal with the focus on not significant studies. Yeah, this could be. I don't know about a specific journal, maybe you know, but I think that there are efforts being made to go against this file drawer problem. So to better have a look at all of the studies that are being published, also the non-significant ones. And we will also later talk about something called preprints that is a solution to this. If anybody else has any questions, feel free to write them and I'll address them in the next block. Yeah, I can also research it later. I think it's maybe not even only one, but it could be multiple journals who adopt this practice that say, or that, let's say, don't put such a big weight on this study being significant or the study being novel and the best ever. Okay. So I will continue. Great. Okay. Okay, next we'll talk about the open science movement. Yeah, the file drawer that I also have in my slides at the end, so we can have a look at that. Okay, so what is the open science movement? In general, open science is an umbrella term that is used to refer to concepts of openness, transparency, reproducibility, and replicability. So all concepts that are already considered fundamental features of science. So open science proposes reforms to improve all of these concepts like transparency, replicability, and credibility of research. And this is not only relevant for researchers, but also for editors, reviewers, publishers, basically for all people involved in the scientific process. Open science means nothing else as that it wants science to be self-correcting, meaning that scientists can take all information that is available to them, that others, results that other people found, share all of this so everybody can use the information that is out there and critically look at the research. So let's say somebody published a study that says that if you put a pen in your mouth and it makes you smile, you are happier. Another scientist reads this and thinks, well, I can't believe it. Let's just try to replicate this. He replicates it and doesn't find the same result. So this is what I mean with self-correcting. Another researcher can then publish another finding saying, well, I found the opposite. What about that? And this way, a kind of discourse or a conversation is starting between researchers and between what is being found. And interestingly, everybody or lots of people always describe the open science movement as something new, but to be honest, it's just a return to the core beliefs of science that were already there from the beginning and that kind of got lost a bit on the way. And it is especially possible now due to several different factors, and focusing on psychology. Especially in psychology, we have a lot of insight already into solutions and, for example, also into the biases that can be introduced when we do research, like the hindsight bias that could lead to harking, so that we say, we thought of this already in the beginning, and this does not necessarily need to happen consciously. So we can also just unconsciously feel like, yeah, we had this idea in the beginning and now it's significant, so perfect. This is what we thought all along. So I think psychology as a discipline can already contribute quite a bit to the different factors that can influence science. A second point is that the problem is very general. So it doesn't only exist in psychology. but it's, for example, also very strong in the medical discipline. So it's something that should not be ignored just because every research field seems to have it. Of course, nowadays, we have a lot more possibilities regarding technology. So with the amount of data that we are processing, the amount of data that we want to share, we can publish supplements online with much more information. We have servers where we can store big data. We can share data much more easily. And this was not possible, or not possible as much a few years ago, but now it's becoming more and more that it is made easier for scientists to share data and to get data. And lastly, the demographics are also changing a bit, meaning that early career researchers, so younger researchers that are just starting out, might come in contact with open science just because they are starting now, and they might have a higher motivation to share their data openly, to get a more extensive training in different labs, compared to older, more established researchers who, let's say, did it always like this, and therefore it's harder for them to change. But that doesn't mean that there are well-established researchers in the field who are not open to the reforms that open science proposes. I'm just saying that it could be harder for them to change the status quo, let's say the way that they always did science, to this movement that is now emerging more and more, and to change it. So here's just a short slide why you should care, because I mean, you're a bachelor student, so you're maybe still relatively far away from doing research independently, for example, as a PhD. But still, it's already relevant for you now. For example, because you are the future generation of researchers in science. So even if you go into other directions, for example, in psychology, it's common to go into therapy, psychotherapy, you still need research. So applied fields, or let's say different therapies, are also based on research. So it is in your best interest that the research that is being done is good research. And then also studies have been done that show that already undergraduates profit from knowledge of this problem, the replication crisis and all of the problems we talked about already, and from knowing possible solutions. And when I first got to know about open science, I felt that I started reading papers much more critically. So I looked at them from a completely different perspective, knowing all of this background information. And I think that's already the best and the only needed reason why you should concern yourself with open science now. Okay, so now I have a short, let's say, why open science or why not open science for you. So of course, there are a lot of disadvantages to open science. For example, many people who are against open science say that it changes the status quo, it shifts norms that are already well established, and we're working fine, so why should we change it? But as you heard now in all of the information I gave you before, maybe nothing is fine, and maybe we do need to change. Lots of researchers also fear for the reputation of psychology or reputation of a single researcher, because, like, imagine now lots of studies are being published that will not be that we cannot replicate. Will not everybody look at psychology and say, well, what are they even doing if nothing that they're doing can be replicated? Or if a single researcher publishes a lot of results online and it turns out that maybe he did some questionable research practices, this will put the reputation at risk. But I think it should. Another reason why not open science is that sharing all of this data and writing the papers, let's say, in a way that everybody knows what is being done and what has been done, sharing your analysis script, can be very inconvenient, and it's a lot of effort, and maybe we don't have time for that. Especially early career researchers are also, or could be, worried that their ideas are being scooped, meaning that if they, for example, tell the research community already very early about the study that they want to do, and they say what hypothesis they have to be able to be transparent, it could be that other people steal their ideas. Although I think it's not really a reason why not, because if you tell the research community, I will do this research openly, everybody knows this, and how should everybody steal your idea then if you already made it public, so to say. And we will see later how we can do that. Another reason that is, I mentioned already a bit, is that it slows down scientific discovery, because if we do all of these replications, we don't discover anything new anymore, because we're just going over and over on with things that we already did, and this wastes time. But actually, it's a good thing with seeing all of these failed replications and this crisis that we have now, to look at the results that we already have. And while we're doing that, we can still discover new things. So I would say that this is also not a good enough reason. For many PhDs that are just starting out now and that are coming in contact with this concept of open science, they sometimes have to choose between doing good research, so really trying to adopt these open science principles, and then having a career. Because as I said, the scientific community still values that you have a lot of publications, you have a lot of fancy findings, and this kind of contradicts itself. So I understand that many researchers are doubting whether they should go in this direction. And finally, people could say that if we say what we're going to do before we do it, we don't have any freedom in data analysis anymore. We can't explore anymore, because we always have to say, this is our hypothesis, then we test it, and then we report what comes out. But this is not true. I mean, all that open science wants is that you are transparent about what you do. You can do what you want, basically, but you just have to be honest about it. Another reason, finally, is that no one will understand anything that we, like, for example, if we upload a lot of materials, a lot of code, it's much too complex. Nobody will care. Well, it could be, but there could be this one researcher that really cares and that really wants to know what you did and how you did it. So, as I said, I think that's also not a good enough reason. And the reason that no one needs to understand how the study was done or how the analysis was done is also really, I mean, everybody who is in research and in a certain field is interested how the research is being done. So now we've heard why not open science. So now I will give you an overview of the reasons, at least some of them, why open science. So I really like this quote. It's very easy, but it's, as I said also in my title, it's just good science. It's transparent science that helps everybody get all the information that they need and to take their own opinions about it. With open science, you are able to disseminate, publish reliable information, and importantly, all of the information that you have. This way, you're able to increase the faith in the research that you do, and you also have more faith in the research that other people do. While at the same time, not keeping knowledge in, but giving it to everyone. In a way, we're also saving resources, because if we publish our data, other people can use it and build on the work that we have already done, rather than making the same mistakes over and over again. And all of these things ensure that then the readers of the publication can really assess the research and the quality of the work because they have all of the information. Good. So we have another short break, and I check if there's anything in the chat. If anybody has any questions, please feel free to write them. Otherwise, we'll just go on and check in the next, after the next part. Good. So now we will talk about some solutions that open science offers for all of these problems that we talked about. So in general, there are a lot of guidelines that are being published about open science principles. One is that research should be fair, meaning that it should be findable. So it should be online. It should have a DOI in this case, or it should be somewhere published online. And this talks about the file drawer problem. So research should be accessible for anyone. That's also already the second point. So it should not only be findable, but we should also be able to access it and read it. For example, open source in some online repository, and we will get to that later. It should use a common language, meaning that if we publish something online, we should think about if other people will also understand it. Using abbreviations that nobody understands really is far away from the point of open science. And the last point is that it should be reusable. So what you publish online in a way should be organized, labeled, described in a way that other people can take it and can do their study the same way, or can use it for themselves, for their own research. So this is for the researchers. And then there are also a lot of guidelines from journals or for journals that they can adopt. And then they can ask the people who want to publish their studies in those journals to also adopt those guidelines. And for example, there are these top guidelines. They are called Transparency and Openness Promotion Guidelines. There are 3 levels for each of these points that are listed on the slides. And the journal can say for each of these what level they adopt. So 1 means there's basically no adoption, little or no adoption of this principle. 2 would be that the journal expects a bit stronger that authors adhere to all of these points. And 3 is the strongest standard. That would basically mean that authors have to adhere to these guidelines when they want to publish in this journal. So this is something that the journals can do. This is something that is from a recent preprint, so something that we will also talk about today, preprints. I think it's a very good overview where to begin when you're starting out with open science. And I also put the publication later on the slide so you can have a look, because this is a really nice article about where to begin so how do you start? What are some practical tips? How can you get into open science? And there are a lot of different things you can do in different steps. Again, we have this research cycle or this research workflow, and we will talk about some of them today. So we will talk about pre-registration, registered reports, replication, preprints, and publication badges. And that doesn't mean that the others are less important, but these are the ones that I think are very good to know about and to start out with. So if we look again at the research cycle, we have replications that we will talk about first. After that, we will talk about pre-registrations, that is relevant when you design a study. Then we will talk about registered reports, that is similar to pre-registrations, but a little bit different. And then we will talk about preprints. And finally, we will talk about the publication or open science badges. So let's start with replication. Replication basically means establishing the reliability of previous results. And this again adheres to science as being self-correcting. So we can either do the same analysis, so we reanalyze something that has already been published. We use the same data from this study, we reanalyze it, and we look if the same comes out. Or we use different data, so we collect new data, but we do the same analysis and we check for the concept if the same comes out. Replications can be either close or conceptual. Close means that you follow very closely what the authors in the original study did, like the analysis plan, what exact analysis that they did, and you do exactly that. A conceptual or extended replication means that you can add some other research questions or hypotheses that you have, or you can investigate the phenomenon that the original study investigated in a broader way. So you can open it up a bit more. Importantly, replications are, of course, never exactly the same as the original study. Even if you use the same data and you do the same analysis, it's still different because you are doing it at a later time point. They can never be exactly the same. And nowadays, or at least this is something that is unfortunately happening, if a replication is unsuccessful, so if it cannot replicate, it's often blamed on a faulty method or the way that the authors who tried to replicate did it, instead of thinking that maybe the original finding was unreliable or not replicable. And unfortunately, the incentives for doing replications are also really low because they are considered less original and less novel. And we, for example, we recently had an example where in our lab where a study was rejected because it was using data that had already been published somewhere else, even though it was used in a completely different context. And though this was not a replication, for replications even worse. It's basically looking at something again. And lots of journals or the scientific community favors research that is novel, that has never been done before. How was the study? Oh, I'm just looking at the questions. How was the study rejected? Well, the authors submitted the study to the journal, and then the journal, I think, sent it out to review, and they got some expert opinions back from other people in the field, and then decided, well, because the data or some of the data had already been used before in another publication, it was not novel enough. This is why they rejected it. Another question that has been written in the chat is, are there open science reviewed journals? So I think that if you mean that if there are journals who adhere to open science standards, then I would say yes. As I said before, there are these top guidelines, and there are journals who really adopt these guidelines. So you cannot publish in this journal unless you really, you share all of your data, you share all of your scripts, things like this. But it's not the default, I would say. So you have to specifically look for these journals. Okay. An example of a replication that is also relevant for our lab, for the scan unit, is NARPS. It's called the Neuroimaging Analysis, Replication and Prediction Study. And our lab and actually Lei also participated in this. It was published recently in Nature, right? I think. So this replication was, the goal of this replication was to show how variable the analysis of a single neuroimaging, so fMRI data set can be. And here again, I want to mention researcher degrees of freedom. So the same fMRI data set was analyzed by 70 different working groups. So they could analyze it however they wanted. The important thing was to test a certain hypothesis or more than a few hypotheses. And then, yeah, they reported on 9 precisely defined hypotheses. And interestingly, they found out afterwards, when everybody submitted their results, no 2 groups used the same analysis pipeline. So if we look at if we look at analysis in general, it's already a lot of researcher degrees of freedom. But if we look at fMRI, the way we can analyze brain data has so many different options that it's really, it's crazy how 70 different groups could use different analysis pipelines to analyze these 9 defined hypotheses. For example, this is just to illustrate. One hypothesis was that they find something in the ventromedial prefrontal cortex. So they find increased activity for something. And most of the groups who analyzed the data also found it. So there's this red blob in with the red circle around it. But there were also part of the groups who found completely the opposite. And I think that it's a great study to show how important it is to share your data workflow, so your analysis workflow, and how different analysis workflows can come to completely different results. Okay, are there any questions on replication? Otherwise, I will go on to pre-registration, but you can of course also ask them at the end. So no problem, I have the chat window open. Okay, pre-registration is next. If you remember, this is something that we can do when we design a study. So replication is something that comes when a result is already published and we look at the published results. Pre-registration is something that we can do if we are designing a study. So it basically means before we are doing the study, we publicly declare the research that we want to do. And this means that we record the whole research plan prior to data collection, meaning we write a document where we put in all the information what we want to do before we do it. So we put a bit of introduction, we put our hypothesis, very importantly, what we expect. We put down how we want to collect the data, who we will exclude, how we will analyze the data, what we will do if there's missing data. So there are so many researcher degrees of freedom that we can avoid by already putting all of this information there before we do the study. So ahead of time, you commit already to your hypothesis, methods, and your analytic strategy. Pre-registration is something that is very flexible. There are multiple templates online that are either very short or very extensive that you can fill out a lot of questions. Two platforms where you can do that are, for example, As Predicted or the Open Science Framework. And again, the importance of pre-registration is to distinguish between these two types of hypotheses. So because you write down what you expect, you write down your confirmatory hypothesis, but that doesn't mean that you can still explore your data set. You can write in your pre-registration, I expect this and this. About this, I'm not sure, we want to explore the data. It could be like this, but it could be like this. So it's fine if you say that you don't have hypotheses yet, but it's better if you have them to write them down before you start. And this avoids p-hacking, harking. A good thing is that you can also claim early authorship for your ideas. So if we go back to the problem of scooping or of other people stealing your ideas, if you pre-register, the information that you are doing the study is out there and it has a timestamp. So if anybody is doing the same experiment, it's basically clear that you had this idea before. With pre-registration, you just upload your document on, for example, the Open Science Framework. That means that there is no peer review. Peer review means that other experts in the field review your paper and give you feedback before it is actually published in a journal. With pre-registration, there is no peer review involved, meaning you can write anything you want and then upload it. So this is, I would say, a disadvantage because you don't get feedback from others. But the good thing is that then you have a faster start. You can upload it more quickly because you don't have to wait for all of the feedback. And this is also the biggest difference to registered reports, a concept that we will talk about next. But first, I wanted to quickly show you an example of my own pre-registration. So far, I pre-registered 3 of my studies, and this is I think the second one. So I already learned a lot in the first one. I made lots of mistakes, and this I also wanted to say with pre-registration, you cannot put too much information. So if you miss something, it's fine. I mean, just start out, try to write down everything that you think is relevant, and you can always improve. So it's better to do a, let's say, bad pre-registration the first time than to not do one at all. So just to give you a quick overview, I was interested in the concept of placebo analgesia, meaning that if you give someone a pill presented as a painkiller, it will downregulate your own pain. So you will feel less pain if you get a pill that you think is a painkiller. And placebo analgesia, so this concept has been shown to reduce not only your own pain, but also empathy for another person's pain. And this is what I'm investigating in my PhD. And since there are lots of studies out there who found that empathy is positively correlated with pro-social behavior, I was wondering whether placebo analgesia can also change pro-social behavior. So whether when we feel less pain, we are not only less empathetic, but we also help less. So I used a pretty extensive template. This is just an excerpt from it. Here I wrote down my hypothesis. So I had different hypotheses. My first one was that placebo leads to lower empathy. And the second one was that the placebo would also lead to lower pro-social behavior. So you can be very extensive. You can write down every possible detail, like here I wrote down the sample size, how I determined them, what I will do with dropouts, so people who stop the study, and so on. But you can also keep it really short. For example, this is a whole pre-registration that I found when I was trying to look for a super short pre-registration. And the authors here are basically just pre-registering a certain part of the analysis. So this is also possible. You don't need to pre-register a whole study. You can pre-register an analysis plan. And for example, the Open Science Framework gives you options where you can specify at what point you are in the data collection. So for me, as a take-home message from pre-registration, it's about transparency. It's about saying what you're doing, what you wanted to do before you do it. The contrast to pre-registrations that are very similar, but a bit different, as I said, are registered reports. And if you remember, I said that the pre-registrations can just be uploaded by you when you want. You can write whatever you want and then just upload it on one of these repositories, while registered reports are a bit different. So they are also a pre-registration in a way, meaning that you say what you want to do before you do it. But the difference is that you submit the pre-registration or this registered report to a journal. So instead of just uploading it when you want, you submit it to a journal, and then it is being peer reviewed by others before you start. So this is what I mean, but it takes a bit longer because other people from the field will actually have a look at your pre-registration and give feedback on it. And this is very cool because then you can already get input on your hypothesis, on your background, on what you could maybe improve. And the cool thing is with registered reports is the journal that you submit to, they already provisionally accept your report or your proposed paper, accepted already for publication, no matter what the results will be. Because what you can probably guess is that a pre-registration does not include results yet, because we have not done the study yet. So the journal just judges the scientific merit of the study based on the introduction and the hypothesis and the methods. No matter what the results will be, whether they will be significant or not significant, the study will be published. The cool thing is that you already get input and reviews before you start, so you can actually change some stuff before you start, and maybe this way even improve the study. So the results are unknown when the decision to publish is made, and it avoids a lot of the questionable research practices that I talked about already. This is what a typical process looks like. So there are typically two stages. In stage one, your pre-registration is reviewed, you get some input, and then you get this acceptance, this provisional acceptance, and then you do your study. And then you write the results in the discussion. So you just add the last part and it's reviewed again. And the important thing is just that you follow the protocol. So you have acceptance of publication, you will get published if you follow the protocol that you pre-registered. So it's a bit more strict, I would say, compared to pre-registration, where you can write what you want, publish it online, although you also have to adhere to what you wrote. But here somebody else tells you. Okay, are there any questions on registered reports or pre-registrations? Just write them in the chat. Yeah. So the question was whether pre-registrations or registered reports wouldn't encourage a different kind of peer hacking or altering the information. So of course, if you can also upload a pre-registration after you did a study and then say, I'm just going to do the study now, and then you already did it, but nobody knows. And of course, that is again bad science. So pre-registration is not a solution to all of the problems that we have, but it's also, for me, it feels like a solution that researchers can do to protect themselves. So if you are afraid that you might be you might later engage in peer hacking or in this bias where you say, well, I wanted to research this all along, the result that I found was what I wanted. you can go against that by pre-registering. Yeah, and this is maybe explain why you think it makes it more difficult. So I think that pre-registration it doesn't solve the problem completely, but it is a very good solution to try to tackle the problem from the beginning. One of the questions was, can you also do both a pre-registration and a registered report? So I would say it only makes sense to do one for a certain study, just because they are pretty similar. One is you already fix yourself with a journal, so you already choose a journal, while if you pre-register, you can afterwards try to submit it at any journal that you like, that you think is fine. So I think both of those have advantages and disadvantages, but both of those have the goal of saying what you want to do before you do it. So I would say decide for either. And I really want to do a registered report. Now that I've tried pre-registration, I really want to see the other side of it. Okay, so I think the second to last one that we will talk about are preprints. Preprints are author-created versions of a paper that are not published in a journal yet or have undergone peer review, but that are commonly uploaded when you submit to a journal. So for example, I recently submitted my first paper for my PhD. I submitted it to a journal, but I also at the same time uploaded it as a preprint to a freely accessible online repository. For example, bioRxiv or SciArchive for us. The cool thing is that my study is out immediately. So instead of waiting, let's say 6 months to go to the whole peer review and publication process of a journal, I can immediately show everyone the research that I did and that is finished. So people can access it, people can read it, people can cite it already. It has a DOI, so it's findable online, it's accessible online, and people can share it. So yeah, Lei said that 6 months time is short. It was just an example, but it can take, I mean, it can go super fast, the whole publication process, but it can also take forever. I think even years for some, but maybe that's an extreme example. So this is a solution to the final draw problem that we talked about, because people also, if they have null results, so non-significant results that they maybe will not be easy to publish in a journal, can upload the preprint anyway, because this is free, everybody can do it. Everybody can access the study, and it's also findable. So let's say that a researcher was interested in finding all of the studies about a certain topic. He or she can also include preprints, and this way get a much bigger scope of all of the research that has been done on this topic, instead as if he or she would just use the published papers. The cool thing about preprints is that you can also add updates. So of course, the version that I uploaded now to the preprint server is let's say, the version that I submitted. But if later I have to change something because I get feedback from the reviewers, I can also add updates. And this way you can kind of track the development of a manuscript or of a paper. Here's an example preprint from Lei and from our lab that was published in 2019. And this is also what I think it's SciArchive looks like. So you have the PDF, you can have a look, you can comment it, you can share it, and already make your research accessible to everyone. Okay, the last thing we will talk about as an open science solution is the open science badges. And this is, I think, a relatively new incentive for researchers to share for researchers who share data, materials, or who pre-registered. Because you can do it, but so far you didn't really get the open recognition for it. Well, you did it, and then maybe you mention it in your methods, but the badges are a really nice solution, I think, to make open science principles more open to everyone, so more visible. Yeah, so your efforts of what you did in the open science domain are publicly recognized. And there have already been studies done that the use of these badges correlated to even more increased data sharing. So by showing these badges to people, and a researcher sees that another researcher has this or gets this, you can get these badges, this actually leads to an increase in open science principles such as data sharing. And there are so far, I think, 3 badges. One is pre-registered. That basically means that you pre-registered your study. One is open data. That means you submitted your raw data or some kind of data together with the paper, so people can download it and look at your data and maybe replicate you. And the third badge is open materials. This, for example, can be related to questionnaires that you used or stimuli, the pictures that you showed your participants in your study, all of the materials that you used. So other researchers can also take those and use those in their own studies. Okay, I mean, I'm looking at the chat window anyway, so if there are any questions, this is the last chance to write them in the chat. So now we can have a look again at the oh, this is not, I was gonna get the, but I don't think I have the questions anymore, how you answered them. Well, that's sad. Well, okay, let's just look at each of them in turn. So I asked you whether,
wait, I can share.
Do you have it?
Yeah.
Ah, great, because I don't see it anymore. I think it
okay.
closed.
So if you could stop sharing, and I'll do it.
Yeah. That's great.
Nice. And see?
Yeah.
Let me make it bigger. Yes. Go ahead.
Ah, but wait. I mean, do you have the poll?
Ah, oh wait. Okay.
You know?
Yes.
Yeah. This one. Perfect. Ah,
okay. Sorry. So wait. Okay. So I do not have to share the screen. Okay. Sorry.
No, you don't. I think you should see the.
Oh, everyone sees the results, I guess, right?
Exactly. Now you see. Okay.
Yeah,
so
yeah,
I'll just let's just go through them. So
yeah, go through it. Yeah. Sorry.
So the first one was well, the general question was, do you think this is acceptable scientific behavior? And so adding participants until the results become significant, most of you said no. We had some yes and maybes in there, but I would say this is not acceptable scientific behavior. The second one is excluding participants post hoc to make the results significant. Also, most of you were on the no front, which I think is a good answer. I mean, the maybe I could understand, because I mean, you can exclude participants, but again, the important thing is here that you are transparent about it, so that you say that you excluded them and why you excluded them. Doing some exploratory analysis before the data collection is finished. I mean, this is pretty mixed, and most of the answers, so 40% of you said yes, and I think that is completely fine to do exploratory analysis, again, under the assumption that we are transparent about it. Stopping the data collection as soon as there is a significant result to be efficient. 95% of you said no, and for me, this would also be the correct answer. There are some exceptions to that. And the Bayesian framework, maybe Lei can talk a bit more about that in another lecture, but the Bayesian framework can maybe offer solutions to this problem regarding sample size and data collection. But let's say in the typical statistical analysis, you should say what sample size you want to collect before, and then just do that sample size. The next one is including age or gender as covariates because other analysis didn't work. This is also pretty mixed from the answers. So most of you said maybe, that's 45%. And I would say it's completely fine if you include things like age or gender, because other analysis didn't reveal a result. Again, be transparent about it and say, we found something with age or we found some with gender, but we did not find something if we didn't include those. Measuring many different questionnaires and report those that worked. Yeah, 80% of users said this is not acceptable, and I would also say it's not acceptable. Trying different statistical tests to see which one has the best result. This is very mixed, interestingly. I would say this is not good. You should choose a test and then do it, and not try out different tests and p-hack your way to a significant result. And the last one was not reporting some parts of the study that didn't work out. Yeah, 75% of you said no. We have some maybes, but in general, I would say the goal of open science is to be transparent. And if you did some stuff that didn't work out, just report it as it is. Okay, so I close the poll. And then I share my screen again. Okay, well, now we can go through this quickly because we just did it. Good. So my last point is how you as students can become active now, because I can imagine it's quite, maybe it's still quite far away. I mean, you're getting to know science, you're doing your first analysis and you're looking at your first hypothesis, but still it might still be far away from you actually going into research and doing research and using open science. But there are some options that you can have already now. There are some really cool books that I'm recommending. So the 7 Deadly Sins of Psychology are really good. And there's another book by Christensen et al. That is about transparent and reproducible research. And then there are 2 preprints. If you remember, those are not peer-reviewed, so they're not published yet, but they are already available on PsycArchive, so on an open repository. And one is 7 Easy Steps to Open Science, a reading list that has a lot more information and more links you can have a look at. And a recent preprint that I found, and then I think is really nice, especially if you're a younger student and you're just starting out, is called Easing into Open Science, a tutorial for graduate students. And I mean, graduate students could be PhDs, but I think it's a very nice paper to start reading if you are interested in it. Good. In general, I suggest that you, if you are interested, start informing yourself about the concepts of open science more early on. As I said before, I read papers much more critically now. So if you read a paper, evaluate them based on open science principles, what you know. Look at the methods critically, look at how much data they shared, look at the analysis that they did, if they reported everything. Here's a cool link about the science of the scientific process that is also related to open science, which is called meta science, if you're interested. Here you can check out some pre-registration templates, and you can play around with the P-Hacker app that we did. And in general, stay involved in the community. So there are a lot of people on Twitter you can follow that are into open science a lot. There's the Psych File Drawer, which is a website that publishes null findings or findings that cannot be published. There's Retraction Watch, which is a website that monitors if papers are retracted due to, for example, bad science. There are different blogs and podcasts that you can have a look at if you want to. Yeah, and if there are any other questions, just write them in the chat now. You can also write me an email anytime or follow me on Twitter. I think I had one question. Open science seems a little decentralized. What is the go-to place to start the open science process? I think that's a really good, but a really difficult question. Because open science is kind of a grassroots network, that means it starts in many different places in the world, and everybody seems to push it a little bit further. There's not really one place, but I think that I put some links on there that you can start, some people you can start out, that you can have a look at to get involved and to get to know more about open science. And for example, one way is to look at your own university, if there are some open science seminars or things that are being done in the direction of open science that are available that you can do. But other than that, I think it's trying it out yourself, doing it yourself, and finding what resources are best for you. If there are any other questions, just pop them now. Other than that, I thank you all for your attention and for the very interesting questions that you asked. I think it's always very nice when I also get some questions. And I hope that now you know all a bit more about open science, and you think about it when you read the next paper. Thank you.